What is that factor, Jazz? Genes that code to the big gene code. Oh, what is how do you pronounce the gene codes for building like sex?
Pili or pili. Some people say pili. I've always said pili.
You're supposed to get double L, turn into I.
Or yeah. So that's cytoplasmic.
What? What did you want me to say to that? Cool.
I'm just saying.
Okay, so we'll get to the modeling activity in a couple minutes, but we're going to talk just a little bit more about plasmids before we get started. We mentioned plasmids yesterday as part of a bacteria's DNA, right? They have a main central chromosome, but they also have these little extra bits of DNA that are found on plasmids, these little extra circles. They can be from 5,000 to 2,000 base pairs, which is not very long for a little loop of DNA, but they do have genes on them. They can have 1 or 2 genes, or they can have up to a few dozen genes on that little loop of DNA. So there is genetic information on there. They do copy themselves just like the big circular chromosome does, so they get passed on to the baby bacteria as bacteria go through binary fission. And these plasmids are also the pieces of DNA that are most easily exchanged between one bacteria and another. And we talked about a couple ways that that can happen yesterday. That process of conjugation, that's the F factor that you might have just been looking at in your notes. The conjugation process is when that cytoplasmic bridge forms between 2 bacteria, and these little plasmids can actually just float from one bacteria into another and bring new genes into a bacteria that might not have had those genes before. These can also be taken up from the environment through that process of transformation. Yesterday we talked about how bacteria have specialized proteins that'll just kind of pick up any genetic material that's sitting in their environment. So if any of these little plasmids happen to be hanging out around the bacteria, they'll just kind of grab them and pull them in. And once they've taken in those plasmids, however they do it, they start reading that DNA and transcribing and translating it to make whatever proteins or whatever is coded for on that gene. So they start expressing those genetic instructions just like any other part of their genome. The reason we're focusing on plasmids specifically today is that plasmids are really important in biotechnology because scientists have found that since these little bits of DNA are so easy to transfer from one bacteria to the next, we can modify these plasmids. If there's a gene that we want to give to a bacteria, all we have to do is insert it into a plasmid and then put that plasmid near the bacteria, and the bacteria will grab it and take it in, and then it has that gene that we wanted to give to it. And so doing this, inserting a new gene into a plasmid, we call this making a recombinant plasmid. We use the word recombinant anytime we're kind of combining different bits of genetic information into one strand, like crossing over forms recombinant chromosomes. Here we're inserting a new gene into a plasmid to make a recombinant plasmid. This slide just kind of summarizes basically how this process works. So if you have a plasmid, so up here at the top, we have this little loop of DNA. And first we have to cut that loop with a restriction enzyme. If you remember when we talked about gel electrophoresis before, a restriction enzyme will recognize a specific sequence of DNA and cut the DNA there. So if scientists can find a restriction enzyme that will cut a plasmid at just one spot, they can use it like a little molecular scissors to kind of snip through that plasmid right there. They also, however, need to be able to use that same restriction enzyme to cut the DNA that they want to insert at both ends. So they have to have the same restriction enzyme that'll cut the DNA twice and the plasmid once. And the reason that it has to be the same enzyme is, if you remember, restriction enzymes recognize a specific sequence of DNA bases. And so, say for example, this restriction enzyme, let's see. I'll do a simple one because that'll help my brain. Something like that. If the restriction enzyme recognizes this sequence, some restriction enzymes, when we talked about them before, we talked about them cutting just kind of straight through the DNA strand. Some restriction enzymes do that, and they call that a blunt end to the DNA because it's just flat. But some restriction enzymes will cut kind of like this. So it might go through here and cut here and cut here, which leaves something called a sticky end, because then if we take this apart, one of our pieces of DNA, I'm going to have to erase this and write it again. See if I can remember what I just wrote. One of our pieces of DNA would look like this. Is that what I wrote? I think. And the other piece would look like this. And then I think I had that. If we cut all of these things with the same restriction enzyme, that means that this end of this piece has to be complementary to this piece, right? So these bases want to bond. So that means whatever bases are right here on this piece of DNA are going to want to bond to the bases that are over there. Does that kind of make sense? You will see this in the modeling we're going to do, so if it doesn't totally make sense, it will. But these sticky ends are called that because they want to bond back together. So now we can just take this fragment of DNA and insert it, and it will bond there and there because those bases are complementary to each other, so they will just form their hydrogen bonds. We do need to use the enzyme ligase, if you remember that one from DNA replication. Ligase was the one that kind of came and glued together the sugar phosphate backbone. The enzyme ligase has to come in to kind of glue it together there and there on the backbone of the DNA, and here, that's kind of the final step. But then you have this plasmid that now includes a piece of DNA that you wanted it to have. So you might kind of be thinking, okay, real cool, we can put DNA into bacteria. Why would we ever want to do that? What use does this have? This can be used in a lot of different ways. So this just summarizes a couple of possible uses. One use is if we need a protein, one specific protein, and we need to make lots and lots of it. Remember, once we insert a gene into a plasmid, the bacteria, as it replicates, which bacteria do pretty quickly, it's also going to copy that gene that we gave it. So this is a way that we can make copies of genes in living cells. That's why this last slide said in vivo, as opposed to in vitro, which is in a lab in a glass dish or a test tube. So we can make our own copies of genes using PCR, but this is a way to do it in living cells. And the benefit is that these cells will also read that gene. So say we want to make a protein like human growth hormone or insulin. The fastest way for us to make that protein is to insert that gene, maybe this gene that we added to this vector here, to this recombinant plasmid, is a gene that codes for human growth hormone. And we insert it into bacteria, and these bacteria start reading that gene, and they start making human growth hormone protein. They don't need that protein, but we tell them to make it, so they're making it. Then we can collect all of those bacteria, and we can purify out, we can separate out the human growth hormone, and then we have lots and lots of human growth hormone that we can give to people who need it. We can do the same thing with the insulin. Or a protein that dissolves blood clots. That's another example they gave here. Or we can just research that protein if it's something that we want to do some research on. Sometimes this is also used just to insert the genes themselves. So there's some examples on the left here that the genes can be altered, or the bacteria can be genetically modified a little bit to help clean up toxic waste. There are bacteria that naturally kind of digest oil spills and things like that, and we can kind of enhance that ability and help them survive in different environments so that they can help with things like that. We can just do research on the gene itself. It's another way for us to make lots of copies of the gene. We can also then insert these genes, after we've made a lot of copies of them, into other organisms, like plants, to make them more resistant to a pest or something like this. So there are lots and lots of different uses for making these recombinant plasmids. Questions about this at all? Okay, today we're going to be modeling how this process works. This is something so I mentioned yesterday that we're going to be doing a lab at the beginning of next week where we are going to be transforming bacteria. We are going to give them a piece of DNA in their environment in hopes that the bacteria will take in that piece of DNA. And if they do, they will show a new trait that they didn't have before, so we'll be able to tell if it worked. The piece of DNA that we're giving them is going to be a genetically engineered recombinant plasmid. We don't have the technology to actually make a recombinant plasmid in our classroom, so we're going to model this part with paper. But this is what was done in a laboratory, and we are going to use these recombinant plasmids to try and give them to some bacteria next week. Okay? All right, so when we do this, we need to make a plasmid, we need to make some DNA, and we need some restriction enzymes to use. So we're going to start just by kind of putting together the pieces for this activity. First, grab your blue piece of paper that at the top should say plasmid DNA. One thing that I want you all to notice before you begin cutting, at the very bottom of that paper, there's a little key that tells you what all the different shaded areas mean. Keep that key until you're done with this activity because you'll need to know what that shading means, okay? But the first thing we're going to do is construct a plasmid. So what you need to do is cut apart these long strips of blue paper on the dotted lines. Save that bottom piece. And then you can tape these together in any order. And this is a plasmid, so you're going to tape it into a circle, okay? So you can start cutting those apart. We'll put this all together, and then we'll talk about kind of what's next. Okay, so folks at home, make sure that you save this little bottom part that has the key here. That's going to be important as you answer the questions later. So the first thing you want to do, folks at home, is cut this into strips like this, and then you're going to tape them apart in any order. But so that they eventually will all go together and form a loop. So I'll show you what that looks like here in 1 second. Whoa, that's trippy. Look at that fun thing I just did. All right.
All in the same direction.
Thanks. That's so kind of you. That is so nice. It doesn't matter how they attach.
So as you make the plasmid, the letters should all be going in the same direction, but it doesn't matter what order you put the strips in. Okay, so all your letters need to be pointing the same way, but other than that, it does not matter which order you put the strips together in. But you are making a plasmid, so it should be a circle. So tape them into a big, long strip first, in whatever order you choose. There's tape. Yep, there are a couple rolls of tape floating around. People at home, you should be cutting yours out and taping them together as well.
Yeah, it's okay.
Ruined it for us.
That one is stretched out.
All right, so your plasmid, when it's finished, should be taped in a loop. So the ends need to come together. So the whole thing, when it's finished, should be one big long circle with the letters on the outside. Make sure you save this little piece. You're going to need that later when you're answering questions to make sure you know what everything is. The next thing I'm going to do is take the DNA. And people in class, I'll catch you up in a minute. I'm talking to the people at home so they get the instructions so they don't have to sit here forever. So folks at home, the next thing you're going to do is cut the DNA strands apart. The DNA strands you do need to tape in numerical order to make one big long strand of DNA. So you can see they're numbered at the bottom. So these you'll need to put in order 1, 2, 3, 4, 5, 6 as you tape them together. There's this one up here that I'm using, but I need it just for one more minute. Let me get my DNA together and then somebody can come grab it. Are you guys having a hard time sharing over there or what?
The roll came out.
Oh, like actual.
Like the roll like actually ran out.
Oh, look in my top corner drawer right there. I think there's more in there.
And the
Nope, my desk. Turn around. Turn towards me, Bridget. This corner. Look around in there. I think there's more tape in there. And if not, there's probably some in the bottom. The drawer right below that might have some. Okay, so again, folks at home, as you tape your DNA together, this is strip 1, and then the next one is going to be strip 2 that you'll tape right below it. So these should go in order so that the gene of interest is all next to each other. The DNA strand is not going to get taped into a loop. This is just going to be one big long strand, like, say, a human chromosome.
No, I'm actually not. Oh. Okay.
All right. So when your DNA strand is finished, it should kind of look like this. It's just one big long strand. This shaded section in the middle is what we call the gene of interest. This is the gene that we want to insert into the plasmid that we're making. So your next task, I'm going to explain this to those of you at home, and then I'm going to let you go just work on this for a while, and you can let me know if you have any questions. Your third sheet has a whole bunch of restriction enzymes. It shows you the sequence that each enzyme recognizes and exactly where it would cut through there. And so what you need to do is you need to find a restriction enzyme, and it's going to be a different one depending on how you taped your plasmid together. But you need to find a restriction enzyme that will cut your plasmid in just one place, not more than one because we don't want to destroy the plasmid, but it'll cut your plasmid once, and it will also cut your DNA once above the gene of interest, so somewhere up in this area, and once below the gene of interest, somewhere down here. Okay, so you'll need to kind of look at them and look at the sequence and find an enzyme that works, that meets those criteria. Okay.
So, is it just one enzyme that's in both cells?
There might be more than one. It is possible that there will be a couple that'll work. If you find a couple that'll work, find one that will cut as close as possible to the gene of interest, just so you have less kind of extra DNA. Does that make sense, Emma?
So I'm just like looking at the 9 enzymes, there's one that'll cut
Yes, you're looking for one enzyme that will meet those criteria. Okay, and then you're going to cut your DNA and your plasmid the way that the enzyme tells you. So make sure you follow kind of exactly where the little line on the enzyme is. And then you're going to actually tape the gene of interest in there. There are really specific instructions posted under the homework post about this activity in Classroom. So you can read through those instructions as you work, because I don't want to tell you too much more. It'll get confusing. But you can go ahead and open those instructions. If you want to stay on the meet in case you have questions, you are welcome to do that as you kind of work through the rest of this activity. And then there are some post-activity questions to answer. But if you feel like you understand what you're doing and you want to sign off the meet, just go and finish up this activity and then answer the post-activity questions. You do need to take a couple photographs of your model when you're done so that I can kind of see where you connected your plasmid and your DNA and which restriction enzyme you used and those things. But if you feel like you know what you're doing at this point, you can sign off and read through the instructions in Classroom. And like I said, if you want to just stay on the meet, I'm going to be mostly talking to the people in class and walking them through the next couple steps I just told you. But you can go ahead and work at your own pace now and do this activity at home. Okay? So stick around if you want to. If you want to sign off for the day, that's fine too.
All right, thank you.
Yep.
Is it because it's